So starting with endoplasmic reticulum. Endoplasmic reticulum is a network of interconnecting membranes forming channels or cistern. It is continuous from outer nuclear envelope to the plasma membrane. It appears like railway tracks under an electron microscope. It was identified by George Pallet, who received the Nobel Prize for the same. So this is the picture which is showing the nucleus, and here you see these two structures. These are endoplasmic reticulum, right? You can see the differentiation between these two structures. This is because the endoplasmic reticulum can be differentiated into rough endoplasmic reticulum or smooth endoplasmic reticulum, depending upon whether they have ribosomes present over them or not. The type of endoplasmic reticulum which has ribosomes over it, they are called rough endoplasmic reticulum, whereas the ones which do not have ribosomes over it, they are called smooth endoplasmic reticulum. So these endoplasmic reticulum, they are prominent in protein synthesizing cells. Example, immunoglobulin, that is antibody secreting plasma cells. They are site of synthesis of proteins, glycoproteins and lipoproteins. This is also the picture, another picture which shows the endoplasmic reticulum. It is more clear here. You can see the orange dots. These are ribosomes. So this portion is the rough endoplasmic reticulum, whereas this portion which is devoid of ribosome is smooth endoplasmic reticulum, right? So what is the function of endoplasmic reticulum? See, it is involved in protein synthesis, especially the rough ones, whereas the smooth endoplasmic reticulum is involved in detoxification of the drugs via microsomal cytochrome P450 system, right? It hydroxylates many of the drugs which I mentioned here. So types of endoplasmic reticulum: rough, studded with ribosomes, involved in protein synthesis, whereas smooth endoplasmic reticulum, it lacks ribosomes, involved in lipid synthesis and detoxification. On cell fractionation, endoplasmic reticulum breaks into small vesicles called microsomes. Moving on to Golgi apparatus. What is Golgi apparatus? It is an organelle that was discovered by Camillo Golgi, and it consists of network of flattened smooth membranes and vesicles. It acts as a converging area of the endoplasmic reticulum, right? During protein transport through endoplasmic reticulum, carbohydrate groups are added, forming glycoproteins that move to the Golgi area. So this is the picture showing the Golgi apparatus. This is the cis phase, this part is cis phase, and this is the trans phase of the Golgi apparatus. This is the intermediate portion. This is also called as medial Golgi. See, endoplasmic reticulum synthesizes proteins, right? And the Golgi apparatus is responsible for packaging and sorting and then targeting the proteins to various organelles and various places where the proteins are meant to be delivered, right? So here, this is the cell and this is the nucleus. This is endoplasmic reticulum, right? So endoplasmic reticulum, this is rough endoplasmic reticulum. You can see the ribosomes over it. So here, this is the area where proteins are going to be synthesized, and then after protein formation, they are going to go to the cis or the forming phase of the Golgi apparatus. From cis phase, they're going to go into the medial part, and then modification is going to happen, and they are eventually going to be released from the trans phase or the maturing phase, right? The modification here is basically the signals are being added so that the packaged proteins, they are labeled in such a way so that it can be targeted to various parts where the protein has to be delivered. So this structure, it is composed of cis, medial and transcisternae. The transport pathway is endoplasmic reticulum. From endoplasmic reticulum, it moves to cis Golgi, then to medial Golgi, and then to trans Golgi. What moves? The protein, the vesicles, they move from this direction to this. Trans Golgi is rich in vesicles containing glycoproteins. Sorting and packaging of newly synthesized proteins occur here. It is an energy dependent process, right? The main functions of Golgi apparatus is sorting, packaging and secretion of proteins and lipids. So what are the fates of the Golgi apparatus, Golgi products, sorry. First is continuous secretion. So whatever the proteins that are synthesized from the endoplasmic reticulum, they go into the Golgi apparatus, and then through cis phase, medial phase, and trans phase, they are eventually secreted out of the cell, right? For example, immunoglobulin released by the plasma cells. The second phase is incorporation into the plasma membranes. From the trans phase of the Golgi apparatus, the vesicles, they go, if the signal is for the plasma membrane, then the vesicle, the protein containing vesicle is going to the plasma membrane, and then the products are going to be utilized by the plasma membrane in its structure. So they are not secreted, they are being incorporated into the plasma membrane. Third phase is formation of secretory vesicles. So these are the vesicles which contain the proteins inside them. For example, trypsinogen and insulin, right? So the contents of these vesicles are released on stimulation, right? Trypsinogen, the one which is responsible for digestion of proteins, and insulin helps in glucose absorption, right? The next fate of the Golgi product can be formation of lysosome for intracellular digestion. Moving to the next organelle, that is lysosomes. Lysosomes was discovered by Dewey, and he was also awarded Nobel Prize for the same. What are lysosomes? Lysosomes are basically tiny membrane-bound enzyme-containing organelles. These are called bag of enzymes. They function as a cell's waste disposal system. It is similar to incinerators in the city. So you can see this diagram. This is the lipid bilayer, and inside it there are variety of hydrolytic enzyme, iodine mixture. This is a lysozyme. This is basically a half cut section that is being shown to in the picture. So let us understand what happens actually in lysosome, how this all functions. Okay, so you can see that this is a microbe, and over the microbe there are present some antigens of the microbe, right? So when that microbe enters inside the cell, our body has some antibodies. They bind to the antigens. So this complex, whole of this complex is recognized by this cell, and as a result of which the cell forms a pseudopod-like structure, and it engulfs the microbe by the process of phagocytosis. So by this phagocytosis, it forms a phagosome, right? It forms a phagosome which has this antibody-coated microbe, right? This is the microbe which has antigens over it. Now this phagosome fuses with the lysosome, and it forms a phagolysosome, right? So in this phagolysosome, all the hydrolytic enzymes of the lysosome can act upon it, and it fragments the microbe into smaller portions. And then this eventually results in digestion of the microbe, and all the particles, all the debris is basically expelled from the cell. So this is how antigen presenting cells digest the particles, and then they project the microbial antigens out, right? Which serve as a signal for the other cytotoxic T cells or T helper cells. Okay, next is formation and function. Primary lysosome fuses with endocytic vesicle or phagosome and forms a secondary lysosome, that is digestive vacuole. Foreign particles are digested within these vacuoles, and hydrolyzed products are utilized by the cell. Enzymes act locally when the membrane is intact. Membrane rupture releases enzymes, which leads to tissue damage due to uncontrolled digestion. Membrane rupture releases enzymes, which causes tissue damage due to uncontrolled digestion. Properties of lysosome is that it contains enzyme which function in optimal pH of 5, right? And it the acidic pH is maintained within the lysosome via proton pump. So we know that enzymes, they are highly specific in nature. They require specific pH, temperature, and specific conditions for them to work optimally, right? So hydrolytic enzymes of lysosome, they need an acidic pH. Types of lysosomal enzymes. There are a variety of lysosomal enzymes that are present, which are capable of hydrolyzing polysaccharides and protein and nucleic acid and lipid. All these enzymes are the examples of lysosomal enzymes. Clinical applications of lysozymes. Let's study one by one. First is gout. Gout is a condition in which there is increased uric acid levels and urate crystals are deposited, right? So what happens in gout, that urate crystals are phagocytosed in similar mechanism that I showed you in the picture. So urate crystals are phagocytosed and they cause physical damage to the lysosomes. It causes enzyme release. All the hydrolytic enzymes are released and causes inflammation and arthritis, right? Second is post-mortem autolysis. See, after the cell death, lysosome ruptures, releases hydrolytic enzymes, which results in self-digestion of tissues. This is what happens after the body dies, like after death. The body decomposes, and this is how it decomposes. The lysosomes ruptures, the enzymes begin to digest itself, the tissues, right? So there's self-digestion of tissues, post-mortem autolysis. Thirdly, we have tumor metastasis. Cathepsis, these are lysosomal proteases. They are released outside the cell. They degrade collagen and elastin, which are the connective tissues. So it results in basal lamina destruction. As a result, the tumor spreads. So this is what is called tumor metastasis. What is metastasis? Metastasis is the property of the tumor cells in which they detach from their original place of origin, and then they move and they migrate to some other distant organ or distant place, and they begin to grow and multiply there. That is called metastasis. So this is how it happens. Lysosomes, they basically degrade the connective tissue, providing room for the tumor to spread. Next is lysosomal storage disorders. Genetic deficiencies or absence of lysosomal enzyme causes accumulation of lipid or polysaccharides inside the cells, right? So it causes lysosomal storage disorders. And then we have silicosis. Silicosis is the occupational lung disease. So this is most commonly seen in people who are working in mines and in other factories. So what happens is that inhaled silica is taken up by the phagocytes, right? So the lysosome rupture occurs because of the membrane integrity is disturbed. Enzymes are released. It causes fibroblast proliferation, which results in fibrosis and reduced lung elasticity, right? And then we have inclusion cell disease, that is called I cell disease. Mannose 6-phosphate defect. Enzymes synthesized but fail to reach the lysosomes. Enzymes appear in blood. It's a protein targeting defect. We'll come to know more about it when we'll study protein targeting defect disorders. Okay. Now moving on to the next topic, that is peroxisomes. Peroxisomes are small membrane-bound organelles. They contain granular matrix which is rich in oxidative enzymes, mainly catalase and peroxidase. It is prominent in leukocytes and platelets, right? So what is the function of a peroxisome? It causes, there's peroxidation of polyunsaturated fatty acids, formation of hydroperoxides, right? So catalase and peroxidase present inside the peroxisomes, they neutralize harmful peroxides and free radicals, basically protecting the cells. It helps in detoxification, lipid metabolism, and regulation of the oxidative stress. So this table basically shows various disorders and effects and consequences. Adrenoleukodystrophy, Brown-Schilder's disease, Zellweger syndrome, and primary hyperoxaluria. Right? In ALD, there's deficiency of peroxisomal matrix proteins, which results in defective oxidation of very long-chain fatty acids, and consequently, there is progressive degeneration of liver, kidney, brain. It's a rare autosomal recessive disorder, right? In Zellweger syndrome, there is defective transport of proteins into peroxisomes, and there is formation of empty peroxisomes, which are called peroxisomal ghosts. So it's a protein targeting disorder defect leading to severe neurological and metabolic abnormalities. Then we have primary hyperoxaluria, defective peroxisomal metabolism of oxalate, right? It results in accumulation of oxalate, kidney formation, kidney stone formation, and renal damage. Thank you.